All right, let's get started. So welcome everyone. I'm Allie Keene with the New York Stem Cell Foundation Research Institute. Welcome to our live virtual research update. Thank you for taking the time out of your busy schedules to join us. We do have several other upcoming educational events with more to be added in the coming days. So be sure to keep checking back to NYSCF.org frequently as we continue to add those virtual events. Today we have Dr. Giuseppe Maria de Peppo, NYSCF Ralph Lauren, Principal Investigator, joining us. You will be learning all about his innovative research applying advanced bioengineering methods to create personalized bone grafts out of stem cells. Before I hand things over to Dr. de Peppo, I wanted to let you know a few technical things first. You are already muted, so you do not need to worry about us hearing you or whatever is going on in the background. If you have any issues with the webinar, you can't see or you can't hear, please let us know in the participant chat and we'll try to get that resolved as quickly as possible. We welcome all questions. Please submit them through the Q&A chat located at the bottom of your screen. We will have Dr. de Peppo answer as many questions as possible. We do have a few guests working behind the scenes to help answer questions since we have so many people joining today. They'll be answering some of your questions right in the chat, but we'll have Dr. de Peppo answer as many as possible. Today's webinar is being recorded, and we will be sure to share a link with you all following the event so that you can share it with anyone you think might be interested. I am now happy to introduce Dr. Giuseppe Maria de Peppo.
Hi, everyone. Welcome. Thank you so much for joining this webinar. My name is Giuseppe. I'm from Italy, and I've been with NYSCF for about 9 years. So in a few words, what I do here at NYSCF, I use stem cells to do orthopedic research. So we do many different things, but today I will focus on some specific projects where we use stem cells to engineer personalized bone implants. So why do we need bone implants in the first place? Why this doesn't move? Wait. Oh, okay, sorry. So why do we need the bone implants in the first place? Well, because I mean, the bone is transplanted very often. It's actually the second most transplanted tissue worldwide. And the reasons why the bone is needed are many. One is because, trauma, of course, is one of the first reasons why we need bone implants. And this can be caused by, bullets on the battlefield, or when we crash with cars, or also sometimes athletes, they have injuries to an extent requiring reconstructions. Another reason is because of cancer. And, one of the most common cancer bone cancer in kids is the osteosarcoma. Here you have an example in this image of an osteosarcoma cancer in the humerus of a child. You see, I mean, there is a large area of bone that is diseased. So in this case, you have to remove this piece of bone, the diseased bone, and then you have to use an implant to reconstruct the defect that you have created. Or, for example, chronic infections. One of the most common infections of bone is called osteomyelitis. This can be caused by bacteria or fungi, but often, if it becomes chronic, then, you have to remove the tissue that is infected. And also in this case, you have to use an implant or a transplant to repair the defects that you have created. Last cause of bone defects that require transplants is, of course, bone diseases. There are many different types of bone diseases. Here I have listed some important ones, such as, for example, osteoporosis, osteogenesis imperfecta, and also skeletal dysplasias. So this, you can see here a case of osteogenesis imperfecta. Basically, in these patients, the bones are very fragile, so they tend to, fracture very easily. And, in this case, you have to repair the defects that you have created. How bone transplant, bone defects are treated? Well, first of all, one can, use a transplant. You can take a piece of bone from the same patient and then, use that to repair a defect. But you can imagine that the amount of available bone that you can use from the same patient is very limited. So This can just be used for very small bone defects. Or if the defects are larger and you cannot take the bone from the same patients, then you can use, for example, cadaveric bone. So basically, you take the bone from a cadaver, you remove the cells, and you use it as a piece of decellularized bone, acellularized bone. Now, I mean, this bone doesn't really work as the transplanted bone from the same patient because the cells are not there anymore. So, I mean, the therapeutic efficacy of these grafts, these bone transplants, is a little bit lower. Or you can use, for example, what's called alloplastic materials. These are artificial materials, for example, metals. They're very commonly used for repairing defects, as for example, prosthetic implants or dental implants. And again, you know, this can restore functionality to some extent, but they also come with a lot of limitations because they can create chronic infections. They can create like a kind of severe inflammatory response and so forth. So, I mean, they do not really represent ideal treatments for people suffering from bone defects. One possibility to solve all these problems is to engineer patient-specific bone into the lab using stem cells. And so what's the advantages of creating personalized bone implants from stem cells? First of all, I mean, these bones are living because they are made of stem cells. And because of these, they can, once you implant them into a patient, they can integrate very well with the patient's own body. And for example, in case of kids, where you know, their skeleton is still growing, you cannot use, of course, alloplastic artificial implants because these implants, they do not grow together with the patient's body. And you know, you have to, every probably 1 or 2 years, you have to replace their implant with a new one. So you have to go through a lot of different surgeries, and things can become more complicated than before. Another advantage is that, you know, since you have the cells in these bone implants, of course, I mean, they can help the defect heal much faster. This, you know, again, since the bone that you implant integrates well with the body's, the patient's body, you these tissues, they grow together, they model together with the body, so you don't have to go for a second surgery. Or since you have used the patient cells and this doesn't come from a different donor, you don't have a problem with immune rejection, which is very important when it comes to transplantations. And also no foreign body response. You know, when you put an alloplastic material that is artificial into a body, again, you know, the body can mount a very strong inflammatory response to this material, which, you know, usually ends in the formation of what's called a foreign body response, which can compromise pretty much the treatment. So if you use a bone that is grown from stem cells, you avoid this issue as well. Plus, there is no biomaterial associated infections. You know, I showed you before that you can use these metals to repair bone defects, but often, you know, after a few years, this biomaterial can provoke some infections, and which compromise then again the treatment. But of course, you don't have this if you grow bone implants from stem cells. So, I mean, let me introduce a little bit what stem cells are. Stem cells are very cool cells. And why? Because they display basically two main properties. They can divide, and so you can, you know, make large amounts of cells. And so you need a lot of cells, you know, both for research, because you have to conduct experiments, so you need to have cells as working material, but also for clinical applications, because, you know, sometimes you may need millions, if not billions of cells to treat a patient. So it's very important they can be divided, they can be expanded to large numbers. Plus, and you see here, they are potent, so they have this ability to become all types of cells that make our body. So, I mean, since we are made of cells, and with stem cells, basically, you can treat a large umbrella of diseases, diverse diseases. Now, how we make stem cells on NYSCF? Basically, we take a small biopsies, like a few skin cells from a patient or like a few drops of blood from a patient. And then what we do, we reprogram these cells. When I say reprogram, I mean, we treat these cells with some factors. Specifically, these are proteins that are produced by human embryos. So if you put these molecules inside adult cells or somatic cells, the cells that make our adult body, these cells become again stem cells. Stem cells that are typical are very similar to embryonic stem cells, the stem cells in human embryos that build our body. So of course, I mean, these cells divide very much and they can form all types of cells that make our body. So once we have these stem cells, how we use them on NYSCF? Well, because stem cells divide and can become all types of cells in our body, for example, you can use them for disease modeling. Imagine a patient with a genetic disease, or also a disease you don't know, I mean, which are the underlying causes. Well, you can make stem cells from these patients, and then you can try to understand in the Petri dish what's the causes of the disease, what's wrong with these cells, what's going, what's not really working. Once you understand the causes of the pathologies, then you can also, for example, screen new drugs and see whether they work in a personalized fashion. Because we all know that drugs do not work for everyone the same way. But if you can create stem cells from each individual patient, then you can test all these drugs on the patient-specific stem cells. So you can better understand whether those specific drugs work well with the specific patient. Or, more importantly, you can use these cells that you can create in the laboratory to actually replace or treat people who are suffering from all kinds of diseases, I would say. Now, stem cell therapy is a very broad word. You can use the cells in different ways. For example, you can generate, I don't know, neurons, and then you can infuse these neurons in patients suffering from some neurodegenerative disorder alone. But you can also combine these cells with some other components to actually create large tissues in the laboratory. And in the case, for example, in my lab, we grow bone. Now, this is a schematic that shows how we approach this problem. So how we grow bone implants in the lab. So again, we take the patient-specific cells and then we see them on something that we call scaffold. I'll tell you what scaffold is in a minute. But in a few words, scaffolds are like templates. It's like a framework that support the cells. And it helps you to really shape the tissue that you want, the implant that you want to grow in the lab, so that you can match the patient's defect. And then you use these bioreactors. This is like a kind of next generation Petri dish that really helps, to scale up the size of these implants, at least to a certain extent. And then you use some molecules, that tell the stem cells to become bone cells. At the end of the day, you have a personalized bone, graft, or implant that is produced starting from patient cells that can be used to repair the defects, the bone defects. Now, in particular, we start from stem cells that we make from, we said, like either a few skin cells or a few drops of blood. And then we induce them using some specific molecules into an intermediate cell type, which is called mesenchymal stem cells, right? These cells have different properties, but one of them is that they can actually form bone cells, cartilage cells, and fat cells in the Petri dish. So these cells have been used to engineer, to grow these tissues in the laboratory. Now, these cells are also important because they can release a lot of important factors. They can regulate the immune and the inflammatory response. So these cells alone have also been used to in different types of cell therapies to help treating a lot of different disorders. But I mean, this is not really the topic of this presentation. So we will focus on using mesenchymal stem cells to grow bone implants, right? So now let's go back a little bit. I want to give you a few, some more information on what scaffolds are, because then I'll show you what we did recently, so at least you can follow better. But basically, scaffolds are materials. They provide structural support and also some stimuli. They can be physical or chemical stimuli to the cells. So help the cells to become what you want. For example, if you want to make a bone, then you provide those stimuli that help the cells becoming bone. And also act, as I said before, as template for tissue regeneration. They are also very important because let's assume that I want to grow a piece of bone, a bone implant that is in the shape of a cylinder. How can I tell the cells to organize in the shape? I need this scaffold, which allows the cells to organize in the shape and volume. And there are many different types of scaffolds, of course. I mean, really, really many. They can be of different nature. They can be metals, they can be ceramic material, they can be polymers, they can be composite material made of polymers and cements or so forth. But here I gave you a few examples of scaffolds that we have used in the past. This is a biocoral. This is really a sea creature that is made of calcium carbonate. But it's a composite material made of ceramics and polymers. And you can actually use it. It's been used a lot in Europe to repair bone defects. Then there is a brushite. Brushite is a synthetic material that you make in the lab, starting from calcium and phosphate chemicals. And we did make these scaffolds a few years ago to see whether they could support the formation of bone in the lab. This is instead an example of a 3D printed metallic scaffold that could be used, for example, to, I don't know, reconstruct the scalp. But this is just to give you an example of how scaffolds can be different. But one important thing that usually is common in all scaffolds is that they are porous materials. So they must, you need to have these pores that are interconnected. So when you see the cells on top, the cells can reach inside the volume of this scaffold and populate the entire thing. What bioreactors are? Bioreactors are medical devices that are used to facilitate mass transport, basically to give food to the cells. Because when we grow these tissues in the petri dish, if the tissues are too big, and when I say too big, I mean a few millimeters, well, there is not a vascular system that can deliver the nutrients to the cells, like in our body. So we use these bioreactors as devices that can, help the nutrients circulate and reach the cells inside the implant that you're growing. Plus, they can also provide some physical stimuli. For example, you must know that our bones are very sensitive to mechanical loading. And in fact, when the astronauts go to the outer space, because there's no gravity, their bones start resorbing, become very, very fragile. So, I mean, using this system, we also provide those stimuli so that the cells can feel happy. They are receiving everything they need, and they understand that they have to make bone and stay like that. So, I mean, bioreactors really are helpful if you want to grow bone implants in the lab. Now, a few years ago, when I moved to JAX-NYSCF, we were the first to demonstrate that by using this approach, combining stem cells with scaffolds in bioreactor, we could grow pieces of bone. And what we did actually, after growing these bone implants in a Petri dish, we have put these implants into some animal models. And after 3 months, we have taken the bone implants out and we have studied what happened. So this is like an histological image. Basically, it's like you take your implant and then you make a very thin slice, and then you color this slice with some chemicals to study, how it looks like and what's going on. So basically, what you see that in the very beginning, the tissue was a little bit disorganized, but over time, there are regions where the tissue starts becoming more and more mature, and it really looks like bone, real bone, the native bone that is in our body. Plus, you see that there is the formation, there are blood vessels that are coming, infiltrating the tissue. So as I said before, this is a living piece of bone that is integrating with the host tissue, the patient's body, and it's really becoming again, like fully functional. So, I mean, this was really a big breakthrough that actually we were also mentioned in the Wall Street Journal. Now, the only issue with this is that even though you use bioreactors, and I said before that these bioreactors helps to scale up the size of the implants, the bone implants you grow in the lab. And why? Because they can provide the nutrients, deliver the nutrients to the cells. There is a limit to this. And there is a limit because imagine that this is your scaffold and you put the cells all inside here. At some point, when the cells grow and start dividing, then there is no way to really deliver nutrients anymore. So there is a lit to that. And these are all the things that one must take into consideration. I mean, if you have a large scaffold and which is this porous like kind of template material, it's difficult to see the cells and make sure that the cells, can really go through and populate uniformly the scaffold. Plus, again, the cells divide and then they, build some resistance inside. So you actually cannot use a bioreactor to enable long mass transport. The bioreactors also, another issue is that when you change the shape or the size of these bone implants that you grow in the lab, you have to come up with a new bioreactor design. And this really, it complicates, to transfer the technology, to implement the technology and to allow other people to do it. So, these things really much remain, a lab-made kind of job. Plus, again, since you have a big piece of bone, I mean, in the Petri dish, okay, you have the bioreactor that helps delivering the nutrients, but once you put this implant into a patient body, then since you don't have a bioreactor anymore, plus you don't have these blood vessels and so forth, if it's too big, the piece of tissue, then it can, probably doesn't integrate, it doesn't survive, and then, things do not work. I mean, the treatment is, there's not much to do with that. So to solve all to kind of overcome all the challenges that are associated with the growing large personalized bone implants, we have devised a strategy that we call segmental additive tissue engineering. Basically, in a few words, you can create a three-dimensional model of a bone defect here in red. And then you can partition these in smaller pieces. And then, you can use these smaller pieces as templates to grow bone segments using a bioreactor, okay, that we have designed. I'll show you later in a minute. And, after you grow these bone segments, then you can piece them together so that you can repair these segmental defects, these large defects. Now, I mean, we prepared, in order to make people understand better, what is the technology about, we prepared a video, which lasts about 2 minutes, and I'll show you right now.
Today, when a patient has a missing piece of bone, like a child with osteosarcoma, there are few good options for treatment. At JAX-NYSCF, we will use the power of stem cells to give patients a better option. First, we use advanced imaging techniques to make precise digital 3D models of the parts of a bone that need to be replaced or fixed. Because entire bones cannot be grown in the lab, we break down these digital models into smaller pieces, which we use as guides for growing new bone. We take a skin or blood sample from a patient and turn it into stem cells. We then grow new bone from these stem cells in the exact shape needed with the help of a cell incubator called a bioreactor. Once the bones are finished growing in the lab, the pieces are put back together in the patient using a special bone glue that promotes healing and growth, or with plates and screws, fully repairing the bone. Thanks to philanthropic support, with this revolutionary new research at JAX-NYSCF that did not exist before, we will improve the lives of patients who today have little hope for recovery.
So, right. I mean, I think now it's better understanding with the video. But I want to give you a little bit, some practical example of what we did. Again, we had to come up with a universal design for a bioreactor. So no matter what's the shape and size of the defect, you can always use the same bioreactor. Also because we said before, by designing a bioreactor with the universal design, we could very much facilitate implementation of this technology, because otherwise you always have to come up with a new design, and it takes time and so forth, and things become a little bit more complicated. But we wanted to, design a bioreactor that could work in all cases. So we started with, we came up with an idea about design, and we tested these designs using in silico simulation. I mean, silico simulation, basically what you do, you use these softwares and you do some computer modeling to understand, how the design of the bioreactor may affect the flow, the flow of the medium that delivered the nutrients to the cells. So based on these studies, we were able to come up with this design. This is a bioreactor design rendering that we made in AutoCAD. And also, I mean, the idea was to really build a bioreactor that could be 3D printed. So once you have the design, you could share the design with other physicians as the researchers, and then they could just use that design 3D model to 3D print the bioreactor overnight. And this is how it looks like. So, I mean, this is a bioreactor is made in nylon. Nylon is a material that is compatible with cells and with tissues. And basically what we did here, we tested whether, the network of pipes and channels inside were working. So you see that there is medium, the red thing, coming from the holes at the same time in each chamber. Now here it shows you what we did. In this A here, you have the different scaffolds that are customized. They have specific shapes which corresponds to a specific segment of the bone implant that we want to grow. And then they are placed in some PDMS inserts. PDMS is a plastic that engineers love very much because, it comes in a liquid form, and then you can cure it 60 degrees. So then you can shape it the way you want. So basically, you see that each scaffold fits perfectly inside the PDMS inserts. But the PDMS insert all have a circular shape. So then we place, the scaffolds plus the cells plus these inserts inside our bioreactor, the said bioreactor, with the universal design. And then what we did, we grow this for about 5 weeks in the lab, right? And then when the segments are ready, we check them if the bone has formed. So what you can see here, I mean, these are the different segments, segment 1, 2, 3, 4, 5, 6. We did stain these, what we do, we take the segment and we make again a thin slice, as I showed before, and then we color these slices for using some specific chemicals. To see whether there is new tissue formation, new bone tissue formation. And you see that the pink areas are all the new formed tissue, while the red ones are the scaffolds, the dark red areas. Here we stand for some bone-specific proteins, again, to show that, the tissue is like bone. And for example, osteocalcin, osteopontin, bone cell protein. Now, I mean, I'm not gonna show what we did the same study without a bioreactor. And if you don't use a bioreactor, you cannot form a tissue everywhere. So in the center of these segments, because they are too big, there is no tissue at all. So, I mean, the bioreactors are very important to make sure that you can grow these bone segments. And then, I mean, in addition to serving as clinical, to serving as an implant for repairing bone defects, we are using these bone implants that we grow from stem cells for many other things. So I said before that stem cells can be used to the model diseases. Of course, I mean, we can also use these bone implants to model diseases. Imagine we take the stem cells from a patient with osteoporosis, with osteosarcoma, and then we combine these cells with these bone scaffolds, and then we grow, three-dimensional bone implants. Then again, we can understand how things look like, and also we can test new drugs. We can test new drugs to check if safety, they are safe to that specific patient, and also if they are effective in that specific patients. In addition, we can use these three-dimensional models of bone implants to test bone biomaterials and also dental implants. And I give you now an example of one thing that we're working in all these things, but I show you one of the projects that we just concluded, and where we try to understand how materials work inside the body. So in this case, what we did, we took a model dental implant, a small screw, and then we anchored the screw into a biomimetic scaffold. Again, you see the scaffold has a certain porosity, and then we put the stem cells from the patient onto the scaffold, and then we grew the bone inside the bioreactors for about 7 weeks. Then at the end, we did all the analysis to study the tissue implant interaction process in the Petri dish. Now, in addition to being used as a testing platform to understand, how our body responds to biomaterials, to alloplastic materials, of course, I mean, once you can grow bone surrounding an implant, you could use this as an hybrid implant. Imagine people that has to get, some dental implants, but they don't have enough bone. Then there's no way to put the implant inside the bone because there's no bone. But we could grow, bone surrounding the implants and then use this as an hybrid implant to treat the patients. Now, to sum up what we said before, what we have said during this webinar, it's that the bone implants that we grow from stem cells, of course, have the potential to accelerate orthopedic treatments, but there are many other things that we need to consider. One is that we need to grow these bone implants using protocols that are, compliant with those that are the clinical requirements. For example, often when we grow tissues or stem cells in the lab, we use some animal components, some components that are divided from animal products. And this cannot be done for clinical applications. So we have to try to, adapt our conditions so that we can make things that are safe and can be used in humans. Also, we need to, we grow these tissues, and sometimes it takes weeks or months, and things can go wrong. So we need to develop a real-time monitoring system so that we can guarantee the quality of these products before using them in patients. Also, we have to, once we make them, we need to test the safety and the efficacy of these treatments in animal models of skeletal defects. You first start with small models like mice or rats, but then before moving to humans, you have to test them in larger animals. And usually bone implants are tested in, I don't know, sheep or other animals of the size. And another thing is growing cartilage from stem cells, which is something we are very active on, because at the extremities of the bones, when the bones reach the joints, the bones become cartilage. So in many cases, when you have to repair the defects, the areas that must be repaired is just at the end, at the edge of the bone. So there is some bone and some cartilage. So we're trying to also grow stem cells from cartilage. Now, with this, I conclude. This is probably the most important, slide. And I'd like to thank all the people who have been part of these projects. My team members, Martina, Daria, Asma, and so forth. My collaborators in Sweden, in France, and here in New York. And of course, all the people working on ISF, because without them, all these things wouldn't be possible. But most importantly, I like to thank the Ralph Ricky Lauren Family Foundation for the generous support. Nothing of these things that I have shown today would have been possible without their generous support. Because he has allowed me to really take on these risky and bold projects and to tackle problems from different angles, in a very original ways. So I hope that we can continue working together so that we can really realize the transition of these bone implants from bench to bedside. So with this, I will be happy to answer all questions. And I leave Ali.
Thank you. The presentation was very interesting. Thank you so much. So as I mentioned earlier, please feel free to submit your questions to the Q&A portal. We'll start answering some of those now. So I have a question for you. How long does it take to make a piece of bone in the lab?
So this depends on the size of the piece of bone that you want to grow. This depends on the type of cells that you use. And this depends on kind of optimization studies. You first start and try, 3 weeks. And then if it's not mature enough, you, culture these bone implants for 4 weeks and so forth. So it really takes some time to understand when it's the right time when they are ready to be transplanted or used in patients. In our case, we've been culturing these bone implants for 5 weeks in the Petri dish. So I would say between 1 and 2 months is the time that, it's the time required for having a piece of tissue that is mature enough to have a certain efficacy when you put it in a patient.
Cool, thank you. What is the best material to use as a scaffold?
Well, it depends on the clinical application. As I said before, some people, for example, they have a very large defects in their scalp because of trauma. So in that case, people have used nets of metals combined with cells. They were, 3D printed and so forth. In other cases, for example, if you want to treat a kid whose skeleton or bones are still growing, then you cannot use a piece of metal, of course. So what you want to use a resorbable material. A material that stays there, but after a while, it gets resorbed by the cells and it gets replaced by what the cells produce. So at the end of the day, there is not really difference between the bone implant that you have made from stem cells and the bone that it's inside the body. So things will start growing together. So it really depends on the specific clinical application, and there is no one ideal bone scaffolds for tissue engineering.
Right. So many different things that can be used. So we have a question from Emily. How does the strength and functional recovery in engineered bones compare to intact, non-engineered bones?
Well, I mean, we use scaffolds also to give to these bone implants that we grow from stem cells some good mechanical properties. Okay, so for example, in the case of bone, you need to use scaffolds that are very hard, and that's why people are using metals. We have been using like these composite materials, or we can also use like decellularized bone and so forth. So you need scaffolds that display mechanical properties that match the mechanical properties of the native tissue. So in that sense, when you grow a bone implants in the lab and you have used the right material, your mechanical properties, you know, the ability to withstand the loading and the stretching and so forth, it's very much the same as the native tissue, as the bone that is in our body. So there is another, you know, function of the scaffolds. It's to provide is to help creating a tissue with matching mechanical properties.
Okay. And then we have another question. Could you have off-the-shelf engineered bone, or if someone has an injury, would you have to create their own personalized bone?
This is a very important question because you know, it takes time to build these bones, and once you build them, you also have to make sure that they are good quality. So having a bank of bone implants grown from stem cells would help. The problem is that to make a bank of bone implants made from stem cells, then you have to kind of preserve them. You know, you build them and then you have to preserve it in some ways. And we have tried a few ways to preserve these bone implants that we grow from stem cells. We have put them in the fridge at 4 degrees, or we build them like in the freezer at minus 80 degrees. And we have seen that when you try to store these bone implants, you know, there are some alterations that can vary in terms of how important they are. But of course, I mean, having a bank where, you know, it could, preserving these bone implants could somehow affect the quality of the implants. So the idea is to probably for bone, because it's not very lethal condition when you have bone defects, it would be better to make it fresh. But you know, if you make it fresh, then you have to deliver these bone implants to the patients. For example, we can make the bone implants here in New York, but the patients may be in Australia. So we have identified systems that help delivering these tissues, and by preserving their quality. So I would, you know, make them fresh and then deliver to somewhere else.
Gotcha. So I guess this kind of plays off of that question. Do you envision SATE as an affordable or expensive clinical solution for patients in need of bone transplant or implants?
Well, I have to be honest, tissue engineering, you know, the tissues and that we grow from stem cells today are quite expensive if you would get them. But, you know, we've been putting together a lot of technologies with the objective also to reduce the cost, so that we can make these bone implants available to everyone in the population, not just for the one who can afford them. But again, you know, at this point, things remain quite expensive, I would say. But of course, with further developments in technologies, stem cell technology, biomaterial technology, and so forth, of course, the cost will go down, will drop in the next years.
Well, good. Thank you for answering that question. I know a lot of people probably had that were listening in. So I know we're at 1:15 now. So I'm going to ask Dr. Peppo our closing question that I ask a lot of our researchers. So what makes you excited for the future of stem cell research, your research, whichever one you want to answer most?
Well, life is beautiful, especially if we are healthy. And I can tell you, we look like very complex as individuals, but we are all made of cells and a glue that the cells produce. So this is what makes up our bodies. So when we get disease, when we age, when we get infection, when we get things that do not, affect our health, it's because the cells do not work properly. So if we can make healthy, functional cells in the body for everyone, and we can use this one to replace the cells that are damaged or diseased or dead in our body, then we can actually, theoretically, we can fix all medical problems. So this makes me very excited because life is beautiful, and to live a very healthy and long life is what everyone wants. So I really bet that in the next few decades, we will be able to realize this promise and use stem cells to make people living better lives.
Well, I think that's the hope of everyone at NYSCF, and hopefully around the world, that's everyone. So thank you, Dr. Peppo, for taking time to teach us all about your current research. I know you're very busy, so we appreciate you taking the time. We hope everyone had a great time this afternoon. Be sure, as I mentioned, to check back to nyscf.org for more upcoming virtual events for students, the general public, and the scientific community. We're adding more and more each week, so check back often. I also wanted to say thank you to the Stavros Niarchos Foundation for making these events possible. We're excited that we can still connect with all of you even while we're virtual. There will be a brief survey right after the webinar ends. We would love to hear your thoughts and feedback. We wish you all a great remainder of your afternoon and week, and we hope to see you all in our virtual world soon. Have a great afternoon. Bye, everybody.